各位贵宾、师长、同学，大家午安！我是蝴蝶兰仪礼大使罗淑菲，非常高兴能够担任今日的司仪。今天的讲座非常荣幸能够邀请到国立成功大学热带植物与微生物研究所高木优玉山学者莅临本校。演讲的主题为 Negative regulation is a major regulator of gene expression in plants。相信借由这次珍贵难得的机会，能让各位收获满满。在讲座开始之前，先向各位介绍今日的与会贵宾，也请各位给予热情的掌声，表示欢迎。首先是主讲人高木优玉山学者，生命科学院黄建成院长。四岛一郎老师，林其然老师，叶专明老师，杨文明老师，赖建成老师，杨俊义老师。黄浩轩老师，王龙奇老师，也感谢生科院的师长同学拨冗参加。在讲座开始之前，有请高木教授上台为本次演讲海报签名。Please come to the stage and sign the posters. 谢谢教授的签名，教授请留步。接下来，为了感谢教授特别来到本校进行演讲，本校特别准备了一面象征最高荣誉的惠蓀琉璃讲座，要致赠给教授。有请院长上台颁发讲座。请看向摄影师，两位请留步。生命科学院也另外准备了精美礼品，有请院长代表致赠。好的，两位请留步。接下来，请生科院的师长们上台与教授一同合影留念。请生科院的师长们一同上台。谢谢各位师长，师长们请回座，院长请留步。接下来，藉由各位的掌声，我们欢迎院长为讲座进行引言。让我们以掌声欢迎院长。
Okay, good afternoon, everyone. And it's our great honor and pleasure to invite Professor Takagi in our college to make this kind of speech. And actually, as you may understand, I have been in Japan for a long time, so it is more easier for me to introduce him in Japanese, but however, I just using English for his introduction. And Professor Takagi, he was in the BS level, he was in Osaka Kyokyu University, and then he took his master's degree in Tokyo University of Fishery. And then he got his PhD in Nagoya University in the field of biology. And then he began his career from the government research officer, and then as the senior research officer and the group leader in the National Institute of Advanced Industrial Science and Technology. And then he became a professor in Saitama University. and then became the Institute of Environmental Science and Technology, the director in Saitama University. And then he became professor, director in Green Biology Research Institute, also in Saitama University. And after he retired, then he became the professor. He came to Taiwan and became the professor in the Institute of Tropical Plant and Microbiology in National Cheng Kung University in Tainan. And I think for his whole career, you will see a lot of award here. So if I go through this, and maybe the time is over. Okay. And however, this is quite impressed me because he is the high-cited researcher for many years. So I think his published paper is very, very popular, and they, contribute a lot for the science. So let's welcome Professor Takagi for today's talk. Thank you.
Thank you, Professor Fan. So it's a nice introduction for me. I'm very honored to have a chance to talk at the Chongqing University, okay, with my old friend, Ye Jianmin. So usually I give a talk with 15 minutes, but so Ye Jianmin asked me or ordered me to talk longer, so around 70 minutes. So please be patient for the 70 minutes. So my name is Masaru Takagi. It's kind of the little bit complicated name. It's kind of it's okay, right? So the Chinese name is Go Mu Yo, not Go Mu Yo, okay, right? So and then, all right. So I have a chance to work in the National Cheng Kung University as a professor, the visiting professor, as from the last May. So and my major is the plant molecular biology and biotechnology. And my research interest is the regulation of the gene expression in plants. And also we are studying the functional analysis of the transcription factors. So many people ask me, what's the transcription factor you are interested in? We are studying all of them, right? So it's kind of the life work, right? So we are studying the transcription, the biotechnology. What is the purpose of the biotechnology in plants? It's kind of very simple, just to increase the productivity or some qualities or the height of the commercial value for the health or something like that, and also reduce the cost. Okay, and then why do we use the biotechnology? Yeah, usually this is a slide for the lecture of the undergraduate graduate student, right? So we are asking why do we use the biotechnology? Its answer is simple: to get the money, right? That's most important, to get the money, right? So that's the purpose, the final purpose of the biotechnology. Okay, biotechnology is just a tool, okay? So then if you the tool, if it's kind of already people use that, okay, we cannot get the new aspect. So for that, what we need is a basic science, to make the good tool, all right? So kind of what we need is the originality and the creativity, okay? Originality and the novelty. That's important. What we are studying, okay? We are studying for that biotechnology, all right? And then today's topic is negative regulation play a pivotal role on the regulation of the gene expression in plants. So now that I realize this is a kind of young people, the young generation doesn't know that this is a dark side, So that's one of the pity. It's already some old style of this one. It's kind of he's a dark side of people. It's kind of negative world, right? So as you know, the plant provides us the essential items that's for our life, such as the oxygen and food, right? And also, plant provides us lots of the items that's our life is more comfortable. So that the plant provides us not only the essential items, but also the many items that our life is more comfortable, such as the flowers. We spend a huge money for that. We cannot eat the flowers, but we spend the money for just the healing, something like that. Okay, so such as the plant activity is called photosynthesis, and the metabolite and the stress response are regulated by the genes, okay, encoded in the genome. Okay. All the function in the plants is regulated by the genes encoded in the genome, right? So then when these genes, when these genes, under which tissue is these genes expressed, such a spatiotemporal expression is regulated by the transcription factors, right? This is the first step to the gene expression is regulated by transcription factors. In Arabidopsis, 26,000 genes is encoded in the Arabidopsis genome. Okay, this 26,000 genes is regulated all the function of the Arabidopsis. Okay, and that's the expression of the spatiotemporal expression of each gene is regulated by the transcription factors. Okay, that means that the Arabidopsis has 2,000 transcription factors. Okay, 2,000 transcription factors regulate each of the spatiotemporal expression and regulated all the functions of the Arabidopsis. So that's if we can not identify the function of the transcription factors, you know, so that we may be possible to regulate all the functions of the Arabidopsis, right? So that's the number of the transcription factors act as the master regulators, and that's the reason that is why the manipulation of the plant treat as a transcription factor is a useful tool for the manipulation of the plant treat. So that's the study the function of each transcription factor is a very important issue. Right, so that's kind of why we study transcription factor is transcription factor regulates multiple genes, and the transcription factor acts the master regulator of various phenotypes, you know, and useful tool for the manipulation of the plant treat. And also the transcription factor is highly conserved among the various plants. Okay. And this is kind of lecture of the graduate student of the transcription factors. Okay, when compare the transcription factors in Arabidopsis and Drosophila and C. elegans, its genome size is very similar, but the number of the transcription factor is around 3 times in plants compared to the Drosophila and the C. elegans. And this color is kind of the variety of the transcription factors. Compared to the Drosophila and C. elegans, plants has lots of various type of the transcription factor. There are so many variations that they use that. Also, they use a variation of the cis element. Compared to for example, the Drosophila and the C. elegans, more than 50% of the transcription factor is zinc finger transcription factors, right? But in Arabidopsis, its plant has just only 10% is zinc finger, right? So something like that. It's such that's only this figure shows that the transcriptional regulation play a very important role in plants compared to the animals. One of the reasons is okay. So these okay. So and the human being also born as a human being, but the plant is kind of developed any time. They developed from the seedling, or they make flowers, and finally they make fruit, something like that. And the differentiation is so much. Okay. And this is kind of the comparison of the transcription factors, family, and the numbers compared to the Arabidopsis and the Poplar. As you can see, the Poplar and the Arabidopsis are very diverse, but it's kind of also in the family, and also the large number of each transcription factor is almost the same, almost completely the same, right? So that's been highly conserved. And this is a comparison with the lice and the Arabidopsis, and these are kind of the kind of the transcription factor is very, very similar. But in our original sativa, rice has a very rice-specific transcription factors. So that's very different. So such a difference. But basically, the number and the variety of the transcription factor is almost same. Okay, what does it mean that a transcription factor is highly conserved in the plants, right? So that means that if you find the nice transcription factor in the model plants, you can easily find out the ortholog and the homolog in another plant. It's very useful for the biotechnology for that. Okay, then, and also the transcription factor is a function as a master regulator. Just one mutation, single mutation, they drastically change the phenotype. Okay, so this is the famous ABC model. You know, ABC model most of the transcription the genes are transcription factors, right? So it's kind of just one mutation directly changes the phenotype, right? So the master regulators. Right. So and also that also means that transcription level play a very central role. But to identify the function of these transcription factors, or not only transcription factors, it's identify the genes. So the loss of function analysis is more critical. Okay. So then we get the knockout mutant, and we can see that if we can get the defective phenotype, it's easy to reduce the function of such genes. So in other cases, more than 98% of the genes has a tDNA tagged line. It's already knocked out. But this is a problem. So only 10% of the knockout lines of the transcription factor genes shown a phenotype, right? So no visible phenotype, all right? So then, yeah, because of she's upset, all right? So there is the presence of the redundant factors, right? The plant has a lot of redundant genes. So that single knockout down doesn't show any phenotype, right? So and also the number of the plants has a very complicated genome composition. In Arabidopsis, in more than 60% is already duplicated. And wheat is a hexaploid, and tobacco is a tetraploid. So in such the genes, single knockout line does not show any phenotype. If you can get to the phenotype, you are lucky, all right? So to overcome such a gene redundancy, so we developed the gene silence system using the transcription repressor, the name the chimeric repressor gene silence technology. It's a CREST system. We developed in 2003. As kind of a useful tool to make a postdoc happy means that we can get a phenotype. Right? So this is a very, very, very simple and surprising. So at around 2000, before you're born, all right? So we are studying the transcription factors, and then usually we believe that transcription factor must be the activator, but the one of the transcription factor, they do not have the activation activity, but has some repression activity. So that we are interested in such a repressor. At that time, no one studied the repressors. So we chop the input, the pieces, and fuse to another DNA binding domain. Try to find the repression domain, right? So finally, we really surprised. Just only 6 amino acids composed of the charged amino acids, leucine, right? X, leucine, has a very strong repression activity, right? So then, we developed when you fuse this 6 amino acid to another DNA binding domain, such a chimeric protein, it has a strong repression activity and suppresses the target gene expression. And also, surprisingly, more than 50% of the Arabidopsis transcription factor contained this the motif in the C-terminal region. Then we developed some new system to the gene analysis. It's named the gene silence system, the CREST system. Okay, so that's simple. There's any transcription factor fused to the repression domain, a 6 amino acid, and transformed to the Arabidopsis. And then the Arabidopsis that express a chimeric repressor suppress the target gene dominantly over the endogenous and the functionally redundant transcription factors, and the result in a loss of function phenotype. Right? So I showed there's something like that. Okay, so the transcription factor repressor, the suppressor target genes switch off. Okay. And then this crest lines can mimic the TF knockout mutant, right? Some of the transcription factors knockout lines, at that time, we already know that. It's 2003, right? So they say that when we express the chimeric repressor, we can mimic the loss of function phenotype. For example, this is PAP1 is anthocyanin biosynthesis transcription factor. When we convert it into repressor, they do not produce any anthocyanin. And this is cup-shaped, right? So I got a cup. But this is cup-shaped. The cup-shaped is kind of scissors to separate the cotyledon. But the mutant of the cup1, cup2, it results in a cup shape. We can mimic the double mutant phenotype of by expressing repressors. And also, this is AP3 is a famous transcription factor. The mutant of the AP3 is the loss of the stamen. In rice, it's called superwoman, just only the female. And the Arabidopsis do not have the petals and also the stamen, all right? So we can mimic that. But what is the big difference? This knockout lines is a double mutant, recessive. But this is the dominant phenotype, just to express the chimeric repressor. We can induce the phenotype. Right? So and also we use the lots of transition factors and get the various interesting phenotype. This is the lignins. Okay, so but this is the lignin and so the biorepressor NST repression domain. There are no lignins. All right, this plant that do not produce any secondary wall. So and also these genes is increase the biomass and just express the repressor, just increase the biomass. Then we found that this transcription factor is regulate apical dominance, try to suppress the apical dominance. But when they lose the apical dominance, they increase the R6 and everywhere, something like that. Or then, okay, this. So then, okay, and also we fused the various plants, and also not only the Arabidopsis, it's kind of rice and the torfesk and the torenia and chrysanthemum and morning glory and tobacco. So we can induce the loss of function phenotype. For example, this is chrysanthemum is hexaploid, right? Hexaploid. So we transformed it to the IN3 repressor. It's got the ethylene insensitive. So usually the chrysanthemum turn to yellow due to the ethylene, but this is ethylene insensitive. So they keep the young, something like that. Its function is also the hexaploid plant. So then our laboratory is studying the functional analysis of the and to try to identify the transcription factor to regulate the various phenotype, okay? And orizasativa and this one. And I think this system is functioning well. So then we decided, okay, this is also the modified the agamas, the transcription factors. Agamas is induced the petals in Arabidopsis. We transformed it to the cyclamen. Cyclamen has just only 5 petals, but we can succeed to make the cyclamen to the rows, something like that. There's 200 to 300 petals continuously produced for the 3 months. Right? Yeah, just okay. So then we decided, this system functions well. So then something the mutant library is useful for the screening of the useful genes. So we decided to produce some chimeric repressor kind of library. So we get around 70,000, 1,700 transcription factors and convert it into the repressor, all of them, and make the transgenic, each of them, and make the kind of the pool for the seed for the screening. Right? And also in the rice has a 500 transcription factor. We selected around 1,000 and make the overexpression and the other repressor lines. Okay, so you know why we selected 1,000. This is a limited of my money. All right. So the budget is we spend a huge money to produce this one. Okay, using this library. So we started screening the transcription factors. That's the first they involved in abiotic stress. Okay. So that's a screening system is very simple. Okay, just you use the seed library and plated it on the agar plate and added the various abiotic stress, right? Usually the repressor is kind of suppress the gene expression. So the usually idea is we will select some hypersensitive one, right? Because of the repressor. But I have some idea. If we can get the tolerance lines, you know. So it's directly used for the biotechnology. So we screen some tolerance lines from the treated by various abiotic stress. You know. So this is wild type, and this is the stress. Okay, we added. It's a 400 millimolar NaCl. It's a seawater is 600. Okay, so usually the regular plant cannot survive that. But the lines that tolerance show the tolerance to the such a stress. This osmotic and freezing and also drought and heat stress. You know. So surprisingly, we get a lot of the transcription factors that's involved in such a tolerance. And maybe we can apply it to another plant because each of them is a different transcription factors, you know, so that we can use that. But okay, so today's talk is a negative regulator. I am very curious. Usually when we express the repressor, we do the microanalysis. We do the lots of the microanalysis, and what is interesting is a large number of the genes is upregulated by expressing the chimeric repressors. And also this is a MIB102, it's a repressor, it's comfort tolerance, it's a high salt tolerance, right? So the plant is grow very normally. So and then we analyze some gene expression related to the high salt tolerance, you know. So for example, the DOREB1 and the ZAT1, the overexpression of the DOREB1 and the ZAT1 is already known to confer the salt tolerance. But such a genes is always upregulated. Then this is other salt. Okay, without the salt, it's still always upregulated, right? But what is interesting is the overexpression of the DOREB1 and the ZAT1, plant doesn't grow well. You know, they show the tolerance, but it doesn't grow well. But this chimeric repressor lines do not show some defective phenotype, right? So then, and also the many genes are upregulated. Okay, it's kind of think about why. There must be the negative regulator under these transcription factors, right? For example, this one. Right? So for example, the MIB102 is kind of the activator of the negative regulator. So, but when we convert the repressors, negative regulators is suppressed, and the down-regulated genes is upregulated. Right? That's okay. That's there must be the loss of the negative regulators. Right? So then, by releasing some such a negative regulator gene expression is induced. Okay, so welcome to the negative world. Okay, so just think about the various phenotype. Okay, physiological response in plants. So we found the number of the physiological response is regulated by the negative regulators. Right? For example, hormone response. I'll show the detail. Okay. Almost all of them, the hormone response is negatively regulated. All right? So hormone does not induce a gene expression. Okay, finally, it's a gene, various hormone that induce a gene expression, but it's not directly induce that. And the rhythm and the banalization, the stress response, and the metabolome, and seed development, and differentiation, and actually bad formation, You know, it's all the physiological response is regulated by negative regulator. Please think about your system, all right? So maybe the way that it's there regulated by negative one, and also in the various step, it's negative regulation is already known. Genome transcription, something like that. In a genome, DNA methylation, histone modification, and deacetylation, is regulated by the gene expression. It's always negatively suppressed, all right? And the transcription level is repression, all right? And the 50% of the transcription factor contain the repression domain, right? So and the nuclear localization is negatively regulated. Right? And also, post-transcription level is mRNA, is a degradation, the microRNA, siRNA, and alternative splicing is regulated the gene expression, right? So, okay, so there's a lot of the microRNA in not only the plants, in the animals also. And also protein level is as a protein degradation, all right? Ubiquination, the smolation, and the phosphorylation, and protein-protein interaction is negatively regulated at plant level. Okay, so then, all right. So it's in the various phenotype is also the regulated by negative regulators. All right. So then, so by releasing the negative regulation, the response are initiated. Also, just the response is releasing the by negative regulator, or just this is used for the lecture of the graduate student. This is hormone response. Gibberellic acid is a DELA proteins interfere the transcription factor bind to the DNA, right? But so the GA is a promoter degradation of the DELA, right? The gibberellic acid do not promote the gene expression, but by promote the degradation of the negative regulators, right? And also oxane is OXIAA is kind of by the interact with the AR transcription factor, they convert into the repressor and suppress the gene expression. But the oxane promote the degradation of the OXIAA, all right? So by degradation, the ARF is released from the repressor and can activate the gene expression, all right? And also the brassionosteroid is a B2 kinase. They inhibit the brassionosteroid transcription factor to transfer the nuclei. But the brassionosteroid, they inactivate the B2 kinase by phosphorylation. And then, so the brassionosteroid related transcription can transfer the nuclei and start the gene expression, right? And also the ethylene is the CTR1 is kind of the MAP kinase, right? So this destabilized the iron 3 transcription factor. It's kind of key transcription factor of the ethylene responsive. And the ethylene inactivate the CTR1 kinase, do not activate the kinase. And then the iron 3 is stabilized and then can start the gene expression, right? So there are lots of the negative regulator the gene expression and also the photosynthesis. No photosynthesis, hormone response. Okay, yeah, I forgot the GA. Okay, that's okay. And also, this is kind of the plants, right? This is common sense. Meristem is home to this one. Right? Why? Why meristem is produced only here? Why? Because of some meristem formation genes expressed here. It's a kind of easy answer. But if we can find this phenomena, right? So we found one of the transmission factors. It's called the repressor of the embryogenesis. Repressor of the somatic embryo. Okay, so that means, okay, this is the transmission factor is activator. When we convert it into the RS1 in the chimeric repressor, huge the repression domain, what happened? Right? They produce the ectopic meristem formation. Okay, and also the highly severe phenotype. There's everywhere a meristem. Right? They produce the meristem everywhere by releasing this the transcription factor, the negative regulators. Right? So that means that this indicates that so plants has some ability to produce the meristem everywhere. Right? But here is kind of only the microRNA against this one is expressed. Only this area, like the cold spot. They take a meristem, right? So then if we remove this one, right? So then another the affixes come out, so something like that, right? So that's when so the meristem formation is not induced, but some negatively regulated, right? Everywhere, like, they have the ability to produce the meristem everywhere, but it's kind of suppressed by the negative regulators, right? And also this one, it's a kind of chimeric repressor, the expression tomato. But there's also also the mutant that produces the fruits without fertilization. Right? Parastonocopy is a tomato. Also, in a grape, okay, we use a lot of the seedless grape. It's very popular now, right? But just treated with gibberellic acid, we can produce the fruits without the fertilization, okay. And then, question is, all right, fertilization is necessary for the seed formation. Okay, this is true. Okay, the tomato and the grape produce fruits without fertilization. What this indicate, right? That means, okay, the plant have the capacity to produce fruits without fertilization. So that's our hypothesis. Plant acquire the fertilization system. Okay, then they suppress the apomictic production. So okay, plant already has some capacity and capability to produce the fruits and seed without fertilization. But the fertilization system that they acquired, because the mating provides lots of advantage, to get the new gene for the survival strategy. Then they stop the apomictic the production. That's our hypothesis. And then what we are focused in the negative regulation is apomix. You know what is apomix? Apomix is some natural phenomena that produce the seed without fertilization. Right? The large number of the plants, more than 400 species, has produced a seed without fertilization, like a raspberry and dandelion and ginger grass. But in Arabidopsis and the rice and any crop do not have the apomix. Right? So the apomix is a system to produce the seed without fertilization. That means the seeds result in a clone. Right? So but usually the pollen and the stem is pollinated, and then they have the hybrid. Right? But the apomix produces a seed, right, without the fertilization. So then, if we can kind of the development of a mixed induced system provide the revolution in the plant breeding. Why? Why? This is in the breeders already. It's kind of common sense, All right. So okay. So because alpha mix is holy grail. I'm not Christian, so I don't know the holy grail, but it's kind of because the apex allows the fixation of the F1 hybrid trait, so it kind of induce the breeding revolution. So F1 hybrid, the word is F1 hybrid. F1 hybrid trait is the kind of increase the productivity more than 20 to 30%. It's just a very specific combination, but it's kind of guaranteed, So then it's corn, maize. So what we use is almost 100% of the F1 hybrid. This is kind of parents, but the F1 hybrid increases the size. Okay, so that the original corn is kind of you can find some horticulture shop the very small the corn. That's the original one. But this F1 hybrid is kind of increase the productivity, right? So then, but what is the problem is we have to cross every year. Right? F1 hybrid, the next generation is kind of the segregated, right? So we have to every year we have to cross one, right? But in a corn, they have the different they have the female flower and the male flower is different, right? So it's easy to the cross one. But in rice or soybean or the wheat, it's almost impossible. But if you can induce the metastellar lines, it's really possible. But it's a cost a lot of time, and also you need a lots of the human power. In China, it's very popular in the rice, the F1 hybrid, because they increase the productivity more than 20%. Right? So if we can induce the apomix here, right, we can fix such the F1 hybrid trait. It's a kind of fixed one. So that we continue as increase the productivity more than 20 to 30%. That's what they call the holy grass. People apomix is called the holy grass. That's the reason. All right, so then, okay, I skip this one. If we can induce the apomix, there's a lot of the advantage of this one. Right, so the F1 hybrid and also the drastic reduction in the breeding time. Usually, when we fix some one trait, we have to backcross more than 10 times. It takes more than 10 years, right? But if we can do the apomix, we can immediately fix such a nice phenotype, right? Next generation. Also, we can use the metastellar lines, all right? So we don't need the pollen. Okay, boys go home, right? So we don't need just a female. We can produce some seed, right? So then, no worry about the diffuse of the gene diffusion, but in the GM plant, So it's promoted to the growth and outside growth for the GM plant, all right? So then. All right, so there's a peak called the holy grass. So more than 40 years, that people try to find some loci of the apomix. Okay, this is the kind of, for example, the ginger grass. They have the lines. That's the apomix lines and not apomix lines. They compare to the genome analysis because they do the sequence. And then many people they find lots of the loci. That's roots of the apomix. But they cannot fail to find the loci that induce the apomix, right? So that's the gene comparison. More than 40 years, the people use that, right? People that are studying the genome, but they cannot find any loci, right? So maybe, sorry, it's like genome comparison analysis failed to identify the genes, right? So that also. So this is a seed, right? So the seed is composed of the endosperm and the embryo, right? So this the embryo and the endosperm is independently regulated. But so for the complete the apomix, we have studied the apomix both in the embryo and the endosperm. So also so far we know that is the egg cell. Egg cell is some of the genes is imprinted, not inactivated in the egg cell. Okay, but it's just activated in the sperm cell. So then when so that's the plant is imprinted. If the egg cell has imprinted genes, they not produce the embryo, but by fertilization, this complemented this one. One was a BBM transcription factor. It's promoted the egg cell division, right? So this is inactivated by the histone modification. That's we know that. So then it replaces the embryo development until fertilization, and the overexpression of the BMM in egg cell is induced the embryo development without fertilization, right? So and then in the central cell is made the endosperm, and the FWA imprinting genes expressed in the central cell, and inactivated the several target genes, So, but these genes are active in the sperm cell. So then when the sperm cell come and the fertilizate, the central cell start the division, something like that. So I said the seed development is negatively suppressed until fertilization. So our hypothesis is this one. This is a no-apomix plant. It's kind of the seed development is suppressed by some imprinting or something like that. And when we fertilize, it's not suppressed, kind of bypass, bypass and started the seed development. Okay, so then we make the hypothesis. Okay, this is not fertilized. Then we express the chimeric repressor and suppress such a negative regulator, they may be induce the embryogenesis and the endospermogenesis. That's our hypothesis. And then we started the screening about this one, right? By using the chimeric repressor. Right? Okay. And first the endosperm. Okay. We transformed the repressor and into it's a male sterile plants. Right? They do not have they don't have the pollen, so they not produce seed. But we transformed to around the 2,000 transcription factor repressors one by one, and screening the line. That's increased the silk, without fertilization. Right? So then we get the several lines that increase the silk without fertilization, and find the 3 transcription factors, 5 transcription factors named elongation silk without pollination. So it's 5 transcription factors. That's the increased ovules without fertilization. Right? So then we use some very tricky system using the KPL single fertilization mutant. Usually the sperm cells interact with the egg cell or the central cell and they make a seed. But this mutant is very strange. Only the one sperm cell interacts with the egg cell or central cell. Okay, so we search some lines. That's only egg cell the fertilized. Okay. Without the central cell fertilized, the egg cell is developed, but stopped in the globular stage, like an orchid, So they grow after the globular stage. Okay. But when some ESP3, the S6 lines, is kind of automatically expanded about ovules, and they have some capacity to host the embryo to the heart stage. But not to the real one. This is double fertilizer. They produce the complete cotyledons, but this is still stopped in the heart stage, but still much more than the progress, right? So then we started the content of the ovules, and then we found this is kind of the fertilizer. Sugar content in ovules. This is fertilizer. Lots of fructose, glucose, sugar, the starch is accumulated. But without the fertilization, it never accumulates that one. But some ESP, the unfertilized, the expanded ovule has some accumulated several of the sugars, right? Sugars and the starch and fructose, something like that. So they have the ESP that may be involved in to provide the nutrient supply, something like that. Then, okay, let's keep on. Okay, we have the across the several and kind of the synergic effect. All right, so then the next is the embryo. Embryo is the most important for the apomix, right? So the natural apomix is kind of divided into 3 types. Right? One is the spotty, aponospocky, and the somatic embryo. Right? This apomix is the most popular, and most of the plant they use this apomix system. This is kind of the skip the meiosis and produce the diploid seed, and the diploid egg cell and automatically it's develop the embryo, right? So and then the other is the aposcopic is a dandelion. It's a Japanese name is a tampopo. They produce another ovules and automatically produce the seed. Now, this mechanism is still complicated. The other is kind of is very popular in the Stolus. Stolus has a lot of the somatic embryo within the mother cells. We don't know why that they produce the somatic embryo. And then produce the multiple embryo, the seedling, So this is the good and also bad. If we cross, So which is the crossed one? We don't know that until we grow that. Anyhow, sorry. So we take we studied the two type of we approach the two type of the apomix. Okay, one is this one. All right. So one is the somatic embryo. So we already have the chimeric pressure that induces the somatic embryo with a very high frequency, right? Almost 100%. They produce the somatic embryo, and they express this is the mother cells. It's kind of the mother cell tissue. It's a diploid tissue. We use this promoter and express the somatic embryo that induce genes, and we succeeded to produce the double endosperm, the seed. Something like that. Usually the embryo seed has only one embryo, but they have the double, something like that. But the efficiency is not so high. We have to select lots of analyzing promoters. And the other is this one, right? This strategy. So we try to find out the transcription factor that skip the meiosis. All right. So first we try to screen this one. All right, this is kind of so far the report, that's MIME, meiosis to insert to meiosis, skip meiosis. The three mutant genes, three genes is involved in the meiosis. And so triple mutant of this one, OSD1, the SPO11, and the REC8, is a result in the diploid spores, diploid spores. All right. So there are the several type of several level of the meiosis, but I just skip this one. But the SRD skip the second division, and then they make they don't divide it into this one, right? So then this N, but still double N, something like that. And then, so then we get the egg cells. But we have to express, we have to divide it automatically, all right? So we need, if we wait the BBM, so we have to pollinate it. It's contaminated with the sperm cell, sperm nuclei. So we try to find some lines that the chimeric pressure that automatically divided the egg cell, And then, sorry. And usually the sperm cell, when the huge sperm cell, it's produce the egg cell division. But we express the chimeric pressure and to try to find the lines that induce the cell division. So then it's called finishing. All right. So now this is final figures. Right? So then for the embryo, we approach the two type. Okay, skip the meiosis and or induce the somatic embryo, and also the automatic endosperm, and produce some apomix lines, and try to transform the soybean in future. And the monocot, and also we use these lines also transform, and then sperm is use the ESP1, and then produce the rice, okay, without the fertilization, and fix the F1 hybrid lines in rice and wheat. That's in our future plan. All right, if you're interested in the biotechnology, the apomix, please come to my lab. All right, thank you for the listening.
Thank you, Professor, for a brilliant presentation. 接下来是问答时间，有请黄院长为我们主持问答环节。
Okay, so now we are going open for the questions. Is there any questions? We thank Professor Takagi's very, very interesting talks. So I think this is a very rare chance to, ask questions.
Okay, thanks, Professor. Thanks for your speeching. And I'm really interested about the CRISP, these techniques. And I'm wondering, because the CRISP basically is modifying the TF of the plant, right? But I think it's a little bit different to like the normal GMO foods. So is this techniques creating the foods is safe to eat or something?
That's a good question. That's a good question. So the question is, so we are kind of transformed to the chimerical repressor, all right? So here is kind of the how to overcome the GM problem, all right? That's the question, right? All right. So that's a good question. It's there are the two answers. The one, so this is a suppression, right? So we can mimic by the genome editing. It's one of the possibility. Already with several of the chimeric repressor expression lines, we mimic we use the genome editing and kill the target genes and also the transcription factors or mutant lines, right? So then we can mimic that the phenotype, right? In Japan, the genome editing is not a GM plant. I don't know Taiwan, but the other answer is this is not usually the GM plant is transformed to the another organs, the genes, right? This is kind of modification of the internal genes, right? But it's just an excuse with the regulation of the GM plant. It's kind of still they stuck that. But so our target usually all right. So this is a kind of the different story. In the GM plant, what we can use is now is not not many one, just only the soybean and the corn, and also the soybean, corn, and cotton. Cotton, we don't eat that. Okay, don't worry about that, right? So and also the canola, right? So there are no rice, no wheat. You know why? It's kind of the soybean. We don't eat that, right? So in the Japanese and also the Taiwanese people eat a lot of the tofu or something like that. It's not a GM plant. Right? Some of them contain the GM plant, but a very, very short amount of the kind of the 0.1% or 0.001% in the commercial. Right? But most of the soybean used for the feed and oil. Right? Also corn. Also, we eat the corn, but it's not the GM plant. So most of the corn is used for the oil and also the feed. Right? So not indirectly, but the people accept that. Right? Something like that, if you can apply this system to the kind of the non-indirect food, it's very possible because the people already accept that. That's a little bit kind of excuse. But the genome editing is kind of the one with a good escape the system. Okay? But it's always that we talk about the GM plant, everybody. But okay, I show that why we use the biotechnology is kind of profit, right? If people believe that this system is very, very useful, okay, they have the way to escape the GM, right? I think so, right? And also we have to think about that, right? Okay, try to make a superfood. Or you don't need to consume here. Just make the system and get a patent and sell to the majors, That's we can get the money from the outside. That's also the strategy. Don't you think so? Right? A good question. Thank you.
Yeah. Yeah.
Hi, Takashi Sensei. I actually interested about the mechanism of this 6 amino acid that become a repressor. I just don't know why that you fuse 6 amino acid that can or any protein interact with this. Do you know any mechanism for this one?
That's good, but that's question I don't know what to get. So it's kind of the people believe that this is they induce the histone deacetylase, right? But because they interact with the topless that protein, okay? People, it's kind of the many phenomena that show that this area is interact with the topless, and the topless mutant involved in histone deacetylase, right? But people doesn't know the indirectly. But many people believe that it's kind of a concept, you know. It's induced the histone deacetylase. But we treated the histone deacetylase inhibitors. Lots of the inhibitors we use that, but we cannot stop the depression, right? But this is probably this the 6 amino acids do not have the enzymatic activity, so maybe recruit some protein. Okay, we try to identify, try to isolate the genes that interact with this one, and we get a result of the kind of histone modification. You know, histone that methylation is involved that, right? But still, I killed the 4 postdoc already. So that's still working about this, and more than 20 years, we're still fighting about this. I'm sorry for that. And also, what is interesting is this 6 amino acid function as a strong repressor in plants, but not its function in a stronger activator in yeast. That's very curious.
Yeah, that's true. And also, only fused in the C-terminal get phenotype or N-terminal also.
You know, so what is, I don't know why this repression domains always find in the C terminal in the transcription factors. It's a 50% of the Arabidopsis transcription factor has this domain, but it's always in C terminal. But we use the N terminal. It's also function. In the middle, some line, some protein is activated. Function, right? So then. I don't know why the C-terminal, they have a C-terminal. Because kind of must be exposed to the protein hold structure, you know. So maybe that's one of the reasons they have the C-terminal, you know.
And how about if you make it become a dimer or triple, can that increase the repression activity?
Yeah, good question. Yes, I used to the 10, and they try to increase the repression activity. That doesn't change at all. So I give up that. Yeah, I don't know why. Just one is enough. But I just primitive the data, so I didn't try so many transcription factors. It's one of the possibility to do that. It's kind of worth to do that.
I'm wondering about, is there any in vitro experiment you already made? In vitro.
In vitro.
Yeah.
In vitro.
For example, the protein and the DNA interaction.
Yeah, so okay, DNA interaction is DNA binding domain, right? So then, in vitro, yeah, we use lots of the transient assay. Transient assay is a function very clearly, right? So and I really wondered whether the transient assay is involved in the genome or not. You know, that's really wondering. It's still working. Very similar level is working. Yeah. Yes, please.
Hello, Professor. I have a question. If a transcription factor has own repression domain, it might act as a negative regulator. But in other cases, where it can act as a positive regulator.
The question is why the repressor acts as a positive regulator? So sorry. Could you help with that?
If a transcription factor has own repression domain,
yeah.
We can act as a positive regulator.
Positive regulator?
Yeah.
It's okay, transcription factor with the repression domain acts as a positive regulator. Okay, that's a good question. You know, so positive is kind of the, okay, so the molecular function of the repressor is suppress the target gene, molecular function. But the phenotype is something that positive, you know. It's kind of it's you know, even in express a repressor, they kind of induce a phenotype, something like that. That's your question, right? Kind of positive regulator, you know. So sometimes the transcription factor is a transcription activator is act as a negative regulator, all right? So that kind of the molecular function and the kind of phenotype is kind of it's not correlated. Is that my answer? Yeah, okay. But now could you check that? So if I can speak the Chinese, I can communicate so much. I'm sorry for that.
问看看，然后我们再请叶老师翻译。
就是如果一个转录因子它有自己的 repression domain， 那它在其他的情况还可以作为一个就是正向的调控吗？
Yeah, okay, so let's discuss it at dinner time. and also the people, if you're interested in I'm in Taiwan, so if you're interested in this library, you know, so it's possible to collaborate with this library. I bring the library to Taiwan, right? So the try to the something screen, you know, so fishing, right? So something nice. And also we can transform. Yeah, also we can transform to the crop, you know. That's possible. But if we can transform, orchid takes 2 years. So.
Is there any questions? I have one question for this. I'm wondering about you mentioned about, for example, GA can release or can can decrease the depressive effect for the negative regulator, right? Yeah. Why? Is there any mechanism for this?
So the hospitalization is by the GA is kind of promote hospitalization and the interaction protein of the DELA, and then the promoted ubiquitination and targeted by the degradation system, right? 26 degradation system, right? So the promoter. Okay, the DELA is promoted by the GA does not directly degrade the DELA, but some hospitalization they produce So kind of release some interact protein, and then it's kind of naked, and the ubiquitination is started. That's the mechanism. Yeah, that's it's very curious. And the hormone induction is kind of regulated by negative. Why the plant use some negative regulator? They use lots of energy, you know. So always, for example, IN3 is always produced and always degraded, you know. Right? So it costs a lot, but the plant use this one. I don't know. I know. I have the answer. You know why? Such a kind of very curious system. Curious. Well, probably this system is kind of response is very quick compared to the started from the gene expression that make the protein some modification. But this system is very quick the response to that because it's already there, like the fireman, you know, always waiting, and then when the telephone comes, they easily started, right? Something like that. Always prepare that. I think so.
Any questions? No? Okay, so we thank Professor Takagi for this kind of very inspiring speech. So please, just thank him.